Hello to all. I am Dr. Upasna, Assistant Professor, College of Agriculture Sciences, Tirthankar Mahavir University, Muradabad. Today's topic is Method of Food Preservation, which come under the course BAG 641, Principle of Food Science and Nutrition. So let's start with the presentation. In this slide, we will discuss about the different types of method of food preservation, use of low temperature, use of high temperature, use of various chemical preservatives, carbonation, and the combination of two or more of the above method, or we can say the hurdle technology. This is the very innovative and new process techniques to preserve food from spoilage, and these techniques basically used by the mm, in today's era by various food industries. So let's start with the first method, that is the use of low temperature. Low temperature basically apply in the food to retard the chemical and enzymatic reactions, or to prevent the food from the chemical and enzymatic reactions. The reducing temperature of food basically retard or stop the growth of microorganism, or we can say inactivate the growth or activities of microorganism in the food. It does not completely remove the microorganism, or it does not destroy the microorganism. It is only used to inactive the uh, activities of microorganism or enzymes. There are the several techniques we are using for the uh, at low temperature. First one is cellular storage temperature. In cellular storage temperature, there is a structure constructed under the ground, or a room underground is developed which is well insulated and well ventilated, in which the temperature is slightly below than the room temperature, or about 15 degrees Celsius temperature is maintained. These are meant for short-term storage only, and for example, root crops, potatoes, apple, these are stored in this cellular storage. Uh, During the winter months, as the temperature is not low enough to prevent the action of many spoiled organisms or the plant enzymes, so to prevent from these type of microorganism, or to prevent the growth of microorganism or the spoilage microorganism to food, these cell storage are constructed, and the root crops like potatoes, apple, these are stored in it at the temperature of 15 degrees Celsius. Next one is refrigerator or chilling temperature, in which the temperature is maintained 0 to 5 degrees Celsius. At the food kept at this temperature, which slow down the microbial activities and chemical changes resulting in spoilage. These are all stopped at this temperature. The mechanical refrigerators or cold storage are used for this purpose. For example, meat, poultry, egg, fish, fresh milk and meat products, fruits, and other perishable products are preserved and stored at this temperature for the shorter period of time, that may be 2 to 7 days. And in this process also, the activities and the growth of microorganism is prevented. These are not destroyed at this temperature. Basically, the use of low temperature, where we can talk about the low temperature, only the growth and activities are retarded, not the microorganism are being destroyed. The last and third one is freezing temperature, in which the temperature is maintained minus 18 to 40 degrees Celsius. Minus 18 to minus 40 degrees Celsius. In this freezing condition, water in food turn into ice. So that makes the water unavailable for the reactions to occur and for microorganism to grow. The moisture which the most perishable foods like poultry, meat, ice creams, milk and milk products, vegetable juices, these are preserved at this temperature. Heating like blanching, these are the destroying of enzyme and enzyme related activities, microorganism related activities, and after these activities the vegetables are stored or frozen, uh, and these are kept for the several months at this temperature. So in use of low temperature, there are three techniques basically: cellular storage temperature, refrigerator temperature, and freezing temperature. Next one is use of high temperature. The use of high temperature, or the use of heat temperature, that basically destroy the microorganisms completely. The first one is pasteurization, in which the temperature kept below at 100 degrees Celsius. Mostly for the pasteurization, the temperature is used 72 degrees Celsius, and food kept for the 15 seconds only. There are several types, but the most common is HTST, high temperature short time. That is most commonly used temperature for the pasteurization of milk products. The pasteurization is the heat treatment which involve the temperature below 100 degrees Celsius, and that killed a part but not all the microorganism present in the food. The other process, that is sterilization, is used for the complete removal of microorganism from the food, and that is selectively used um, for the food, because it also destroys some nutritive value of the food. So pasteurization is used at 72 degrees Celsius. Mainly we are using 72 degrees Celsius temperature, that do not destroy any type of microorganism, but we are used in milk to enhance the value of, or to enhance the taste of milk. That is also used for the preservation of milk products. Next one is boiling. The temperature is at 100 degrees Celsius. Cooking of rice, vegetable, meat, fish, etc. at home is usually done at this temperature. Or boiling the food with water and involve a temperature around 100 degrees Celsius. This is also an activity which kills the microorganism. Third one is scanning. This is the process which is done above the temperature 100 degrees Celsius, or mostly we are using 121 degrees Celsius temperature for the canning process. In this process, food are heated and sealed in a container or a jar to a temperature that destroy all the microorganisms present in the cans or in the utensils, and inactive the enzymes that could be have health hazard or cause spoilage to the food. So next one is use of chemical preservatives. The chemical preservatives are food additives. Food additives are basically those substances which add mm, some color, flavor, and value to the food, but these are not nutritive substances, basically used for the preservative purpose, and which are especially added to prevent the deterioration and decomposition of a food through enzymes, or we can say through microorganisms. So preservation of these type of chemicals uh, uh, lead to the storage of several months of the food products, and these are basically two types. First one is class 1 preservative, and second one is class 2 preservative. Class 1 preservatives are the first one, include that use of sugar, salt, or we can say these are safe for the human health. The use of these class 1 preservatives are um, safe for the human health or safe for consumption. Basically these are use of natural preservatives like sugar and salt. These are natural. Acetic vinegars, alcohol, these are used in like pickles and jam, jellies, and with the use of this we cannot say that the food is unhealthy for us. These are the relatively safe for our consumption. In class 2 preservatives, these are basically the use of chemicals. The second group include the use of benzoic acid, sulfur dioxide, nitrites and nitrates, and a variety of neutralizers, firming agent, bleaching agent, and referred as class 2 preservatives, and these are also considered as relatively safe for human, but also if these are uses uh, in the permissible limit. If we are using these in the permissible doses that do not cause any health hazard, and that is prescribed by the FSSAI, Food Safety and Standard Authority of India, that use of class 2 preservatives should be limited. We have to use class 1 preservative as much as we can, but class 2 preservative must be under some limited doses. Next one is carbonation. Carbonation is the process by which carbon dioxide gas is dissolved in food under some pressure. The main motive of this carbonation is the removal of oxygen from the drink, or we can say in the liquid. So the medium of growth of microorganism uh, is inhibited. Uh, the presence of oxygen uh, improve, or we can say is the main factor of growth of microorganism in any type of food. If we remove oxygen from the food, the bacteria or other microorganism cannot grow this. So to eliminate or to destroy the microorganism from food, we can use carbon dioxide gas in some like beverages, soft drink. It is present in as a neutral preservatives. Carbonation is the adding of carbon dioxide gas to a beverage, which impart the sparkle and tangy taste and preventing spoilage of these drink. Next one is combination of two or more of the above method or hurdle technology. This is the best technology we are using, or preservation industries are using nowadays. The hurdle is the food is defined as the substances or processing step or various preservation factors inhibit the growth of various microorganisms, resulting in the death of microorganism. That is basically the use of several methods to inhibit the growth of microorganism or to kill the microorganism. This is the safest method we are using to eliminate or controlling the growth of pathogens, making the food safe for consumption and extending its shelf life through the application of a combination of various techniques, or we can say technologies and methods. Uh, Higher the hurdle, the greater the effect of food safety. The quantification of various factors in terms of pH, redox potential, water activity, temperature preservation give a successful level of combination of hurdle, which lead to the failure, growth of and death of microorganism. We can understand this with the, as you can see, there is a process drawn in which high temperature, low temperature, water activity, pH and FW, these are written. What is the meaning of that? For example, if we use meat or milk for processing, first of all we give high temperature to milk, like milk is pasteurized at 72 degrees Celsius or 100 degrees Celsius. The first step is this, to kill the, which kill the microorganism. The second step which we are using, that we are storing that pasteurized milk at low temperature. Low temperature, what is done, that create a inactive growth of those microorganisms which are present after that pasteurization, because the pasteurization is the process which do not kill all the microorganisms. Some microorganisms survive after the pasteurization process. For that purpose we are using low temperature technique, that pasteurized milk we stored at low temperature, or we can say we freeze uh, that pasteurized milk. After that process, what we can do, we remove the available moisture in it, so that if we remove the moisture, the microorganism cannot survive in less moisture. After that, removal of that moisture or reduce in water activity will maintain the pH of that pasteurized milk. in first step, which we pasteurize. After that, milk is stored at low temperature. After that, the water activity is reduced, or we want to make powder of that pasteurized milk, so water activity or moisture is removed, so there are less chances to grow the microorganism in that. After that, the pH is maintained, and after that, the heating process is done, or we can say the other processing is done to reduce the survived microorganism. After that, we pack that item or that milk by vacuum or new packaging technique, which remove the all available microorganism in the food. So these are the combination of one or more method for the preserving of food, and that is called the hurdle technology. Hurdle technology means use of one or more method of food preservation to kill the microorganism completely. So that's all for today's presentation. Thank you.